started adding or taking away electrons. And again, doesn't change the atomic number, mess with the protons. It didn't change the atomic mass because we didn't mess with the protons or the neutrons. By adding and taking away an electron, we change the charge of that atom. It becomes positively or negatively charged, depending upon whether we take away an electron or add an electron. That process is ionization. This is going to be a big deal as we go forward throughout this event. So when we finished, we started talking about electrolytes. Electrolytes. Okay, any questions what I just put through the meeting? Yeah. For my purposes, to be a little bit more specific, the electron change. Because if you change the electrons, obviously you're making ions, and those chemicals, those behave differently. So so electrons are what are behave more inside. By the way, that is the first question I asked that review in 2 years. I don't know what happened for 2 years, but the students all know. Okay. Don't you raise your hand. That's you. All right. Anything else? Yeah. Mm-hmm. That is all the word. Not dealing with all those octets. Anything else? All right, electrolytes. Now, I'm a big proponent of looking at a word and trying to figure out what it is. In fact, we always help to understand this word because we are completely new language. When you are picking Spanish or French or German, so many new languages, maybe you're that odd person who says, no, I want to learn English. Okay. But we are doing a whole new scientific slack medical. So sometimes I will spend a second and say, what's that word? And I look at that word electrolyte and I say, oh, that first part, electro. Tells me exactly what this thing is capable of doing. Electro sound like. Bingo. It's going to be able to conduct electricity. So that's kind of a good way to start. Think about it. Electrolytes are molecules that ionize in water. Remember, an ion cannot form if it's not in water. So these are physiologically / medically important molecules that ionize in water. Chemists will say so. So they are solutions that conduct electricity. Now you can add different things to water, right? I mean, you could just think about the kitchen. I can add sugar to my water, or I could add salt to my water. One of those will conduct electricity, one will not. That is showing sugar, glucose, nope, not going to conduct electricity. Whereas sodium chloride will conduct electricity. What's the difference between this and this? Why is this one conducting and that one is not? Partly right. It says it has more molecules.
Salt is ion.
Salt makes ions. Glucose stays as glucose. Doesn't break apart in the water. But sodium chloride separates in the water because we go back to that statement. Sodium chloride is a molecule that ionizes in water. Remember, what is ionize? Makes an ion, becomes an ion. So we now have a positive and a negative charged ion in that water. And if you flip the switch, what is traveling through the wires to brighten the room up in the lights? Electrons. So that's what's needed for this conduction of electricity in part. So what are some of these? Well, we just looked at fluoride. There's also potassium and calcium and magnesium, phosphate, bicarbonate. These are the ones that we are going to revisit, and we will revisit, and we'll go back and look at them again, and you're just going to get sick and tired of talking about them, but they are that important. So if you go and have your blood drawn for whatever purpose, they're going to print out chemical panel. And that chemical panel will list these and some others. Those are the big ones. And eventually, when you get into practice in a medical field, you're going to be concerned. There's something good. Potassium. So, what do? They can conduct electricity. So, what we're going to find is that cells contain a certain concentration, certain amount of some of these ions. And around the cell is going to be a different concentration of some of those ions. And across the cell membrane, we're going to have the movement of ions. We're going to have a little bit of electrical activity going on. This is the way we can put electrodes on your arms, plug you into a machine, and watch what your heart does. EKG, electrocardiogram. Or you can do it on watch. That's so cool when I have to do that. First time I have to do it. But that's what's going on here. Okay? There. We just used the term molecule. It's a molecule. And what's a compound? They're the same or they're different. Turns out they're a little bit. Molecules are two or more atoms bonded together. Two or more atoms bonded together. It means water is molecule, H2O. CO2. What about carbon molecule? Compounds are two or more different atom bonds. Does that sound different? A little bit? I call everything a molecule in this movement. All right? Chemists up there, they'll debate me on that, but we're just going to say it's a molecule. It's just so much easier. But basically, they are made from different atoms. Atoms that are bonded together. That's the point. Now, what does that mean when we say bonded together? It means that, right? It means Sean Connery, Daniel Craig, office. What we mean is that the atoms are going to be held together in one of three different ways. And those bonds are ionic, covalent, and hydrogen. We're going to explore each one of those. By the way, the best James Bond. If I grew up with yes. Okay. So, what do we mean when we say an ionic bond? Well, we've already talked about what ionic bonds are. They're charged atoms. Or charged molecule. And if you are positively charged and your other friend over there is negatively charged, you're attracted to each other. And you are attracted in such a way that you are sharing, you are attracted to each other, and you are kind of loosely associated with each other. Molecules of salt, sodium chloride, are held together because of that charge difference. Positive, negative attract when they're held together. Put them into water, it separates. Remember, it causes ions to form. So, this is attracted to that, or vice versa. That's ionic bond. Now, for us, another very important one is covalent. Now, ionic can be thought of as giving and taking away. Right? I give an electron away or I take away an electron. Covalent bond is like Sesame Street. Everybody cooperates. Okay? Monsters and humans all get along. Okay? So, what's going on? Here's the hydrogen atom, here's the hydrogen atom. One electron each. But a really stable situation would be to have 2 electrons in that shell. That's about all I ever say about this, right? So there's 2 there. Now, which one belong to that and which one belong to that? I don't know. Doesn't matter. Okay? Mark that somehow, and you put them together, and then you broke them apart. That one could end up with that one. Doesn't matter. They're just pieces of energy. And they're floating around each of them. And it makes for a very stable molecule. So stable that that is actually the hydrogen that's in the atmosphere around us. It's not that. It's that. Hydrogen prefers that situation. Now, below is another gas, CO2. And in this case, we have oxygen, carbon, and an oxygen. But this time, oxygen is bringing 2 electrons from that outer slip ring, and carbon is bringing 2. So that's why this is a double bond, because they're not sharing 1 electron each, they're sharing 2 electrons each. And again, if I count 1, 2, 3, 4, 5, 6, 7, 8. 8 around each of them. And they're happier that way. I anthropomorphize my molecules. So this is like that, but with more electrons. Now, which of these, that or that, would be easier to break apart? Okay. Much easier. If you take that salt molecule, put it in water, we know what it's going to do. Break apart the sodium and chloride ion. But when I take a sugar molecule, which is held together by covalent bonds, we already saw, what does it do? Stays intact. These are strong bonds. They don't break very easily, as we'll see. Now, sometimes when we put together the 2 atoms, they will share the electron fully. And that's a carbon-carbon bond. So the 4 electrons here and the 4 electrons there, they're shared equally. Okay, so they're all floating around both of the nuclei, nice and neat. But what if we have a large nucleus like this oxygen with 8 protons to it, and we have a hydrogen attached to it with only 1 proton? I got 8 positives over here. I have 1 positive over here. Where are the electrons going to tend to be? Over here will be a positive. So that means the electrons tend to be here, which means this end of the molecule tends to be negative. And this end of the molecule tends to be positive because there's only one positive charge here. And that's what makes this polar versus non-polar. I could be all silly and say, oh, notice it's red and blue, and that's the state of politics in the United States. I'm polar. But if that helps you remember it, the polar means that they are not equally shared. Still a covalent bond, they're still sharing the electrons, just sharing them a little less equally. You all, if you haven't said it, you know exactly what I'm meaning about sharing, right? You never share equally. Why, when you become a parent, you learn. One kid cuts the cake, and the other kid gets to choose. And that kid's going to figure out, I got to buy equally. Now, why is that such a big deal, this idea of polarity to our molecule? Why is this non-polar polar thing a big deal? Because the solvent of all life is water, and water forms polar. There's our oxygen negative, our hydrogen positive, the other hydrogen positive, slight and positive. So that slight positive attaches to or attracts to another oxygen, and that hydrogen to another oxygen. You get the idea here? Link, 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 link. We're all linked up. That is called a hydrogen bond. Why is that a big deal? Because when I turn on the water, what happens? It didn't have hydrogen bonds, it would spatter everywhere. If you've ever poured acetone, those of you who nails, polish on you and remove the nail polish. Splatters, or take rubbing out and splatter. Why? It doesn't have as many hydrogen bonds. And then you go down and you look at the pond and you see that little water spider, or water walker, and it's running across the top of the water. Why? Hydrogen bonds. Because it has cohesiveness. Water molecules are cohesive. They are attracted to each other, and that creates tension on the surface. Pour water into a glass, is it flat on top or does it curve up the sides? Curves up the sides. That's because it has adhesive properties. It is attracted to and holds on to the surface of life. Oh, and what's my blood? What does blood do? Stays together because it's cohesive. Kind of moves along, attaches to it. What does ice do? Float or sink? Floats, thank goodness, otherwise the fishies would all get squishy every winter. Why is that a big deal? Hydrogen bonds. Or why did it work rather? Hydrogen bonds. Oh, mind blowing. It is that important. Now, the other purpose for hydrogen bonds, we'll see in a couple of weeks, is DNA. Okay. So, why then also are hydrogen bonds important? Because now we have our water molecules oriented to each other, but also to the positive sodiums and the negative chloride ions. And that's how they ionize salt or any other ion. They orient themselves because of that polar molecule. They orient to the ion, and that's what forms ions as well. we go. Hopefully, this is a little refreshing.
Oh, no.
You have some questions, by all means, ask. Email me, send anonymous notes, send me before, I'll answer you in 5 hours. The 3 bond types. Ionic, covalent, strongest. What's the weakest? Hydrogen. How do we know hydrogen is the weakest? Because I put a little heat under it. What happens? Water goes from liquid to. So those hydrogen bonds are breaking as the water that's loose from the bottom of the water. All right. So a little more basic chemistry from the biological, physiological point of view. We're going to talk about mixtures. And you can see across there on my diagram that there are 3 types of mixtures. I know there's A, B, C, D, but C and D are the same mixture. So the first one is A. A is a solution. And a solution is a solute plus a solvent. Forward, a solution is a solute plus a solvent. A solvent, a liquid, in our case, water. Solute, what you add, sugar, sodium chloride. Whatever. But most importantly, a solution is a small solute, very small molecule. More importantly, it dissolves. Can't see it. It's in there, you just can't see it. You don't go to the ocean and go, oh, the salt's floating around in it. But you know it's there when you taste it. You go over to Chick-fil-A and you get your sweet tea. Man, that is some good sweet tea. You don't see the sugar in there. You might ruin your coffee by putting sugar in it. Black coffee. But you don't see it at all. And it's also small enough to pass through artificial membrane. Now that is somewhat big idea, but what it means is that when we are putting fluids through tubing, that will go through it. You're going to filter things. You might have a patient on dialysis down the road. It will not be filtered through the membrane. So a solution is a solvent plus solvent. So the next one then is called a colloid. Colloid. And a colloid has what are called colloidal particles. Well, it means they're bigger. But what else does that mean? They are so big, they don't dissolve. They're kind of, sort of, almost they're in there. This solution, or excuse me, this mixture is what do you think it is? Any body fluid. What is it? So, no one's ever guessed that one before. Mammals making milk. So the protein and the lipids that are part of milk are the colloidal particles. You don't see them, they're in there. Okay. They're suspended in there, if you will, a little bit. Now, if you leave that milk out, you know what's going to happen. This particle will, there they are. Stay. That's what we mean by colloidal. And those will not pass its membranes. Okay, they're too large. So, mixture, colloid. This is the lacquer. I'm going to call it suspension. What do you think the suspension is? Thinking bodily fluid. Blood. So what's in blood? What's the primary thing that you think of in blood? What? Water and what? Iron. But the iron found in. So notice these particles, these cells are going to eventually separate. The suspended particle plus fluid medium, in this case plasma, but they are very large and they will separate. You draw a tube of blood, put it in the correct tube, type of tube rather, and you leave it there for less than an hour, and you'll see cells are at the bottom, fluids at the top. Okay? That's what we mean by suspension. They are going to separate. Little yellow part. They are going to separate. And because colloids had smaller molecules and were filtered, blood is as well. Suspension particle. Right? Pass that through a filter and then trap the cell. Okay. No, not last. Remember this chemical shot. Right? All right, acids and bases. Now, I'm not really going to harp on strict definitions about what is the difference between an acid and a base in terms of the way this is defined. I'm just giving you the definition for what we're meaning. An acid, by definition, is a proton donor. The world function. Remember, that's positive charge. Positive charge. Yeah, molecule. Nucleus. And if you take the electron away from a hydrogen, all you have left is protons. That's it. So that's why you label it like that. That's the proton. And acids such as hydrochloric acid, they break apart so that they are only hydrogen and only chloride ion. And it's a very, 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 I don't know if I can say that, strong acid. You don't put your hand in it. Certainly don't drink it. Very, very hot. Now, you'll notice the water can also be a little bit of a proton ring. In other words, water, H2O, sometimes give away a hydrogen ion in OH. So if an acid is a proton donor, that means a base is a proton acceptor. Where does proton come from? The acid. So ammonia picks up a hydrogen and becomes ammonium. Water can also accept a hydrogen. So both acid or a base situation. Now, this is not a very strong base. This one is very, very strong. Your body produces this, your body produces that. You know your body produces that. You walk in the locker room and go, whoa, what in the world? You know who produces ammonia, you need it. Yellow, let it mellow, brown, push it down. So if the yellow's mellowing, you'll smell the ammonia. So, Your body produces those. Now, we measure acids and bases, and this is the more important key. We measure acids and bases in stream. So we measure the amount of hydrogen in a solution. How much hydrogen is there? A lot or a little? So pH of 7 is neutral. Don't ask why. I cannot tell you why. On a scale from 0 to 14. Why 14? I've never heard anybody give me a good explanation. Why not 100? Would have made more sense. 50's the middle. 14. So pH of 7 is neutral. pH of 1 to 2, very acidic. That's your stomach acid. Oh my gosh, a burn, right? Oh. Get that little laugh, if it gets all the way up into your mouth, you're like, acid. Coming out of the stomach. pH 10 is basic. Magnesium. Oh, you mean so my stomach is going, a lot of acid production, drinking a little milk of magnesium? What does it do? Neutralizes it. That's proton donor. That's proton. Protons go over the milk of magnesium. Neutralize. Your blood, 735 to 74. I do not ask you to remember very many values in this class. I just wanted you to say over and over and over. Your blood pH is 735 to 745. Slightly basic.
My God, please stop.
Cool. That means probably a little acid in there, probably a little base in there, and they mix in their body what you do just. Mostly neutralized. So, liquid drain cleaner, don't drink that either. 14, don't drink battery acid. I'm sure they told you that. Very, very acidic. Right there's my coffee. It's actually a measured. It's actually short. There's your milk. A little acidic. There's utilized. Now, this pH, I mentioned that's how we measure things, and it is log base 10. And this is one of those little math things that I might ask you to do. Not actually do the math, but understand the math. pH is 7. It's log base 10, that means 10 to the 7th power. pH of 5 means 10 to the 5th power. Difference between 7 and 5 is 2. And 10 squared is 10 times 10, 100. The pH of 5 is 100 times more acidic than pH of 7. That's why when you say pH of 1 versus pH of 7, it's really acidic. 1,000,000 times more acidic. We used to work in a factory. We used something called hydrochloric acid, really like hydrochloric acid. And I had to put on a whole bodysuit, glove, mask, respirator, and all that. Just open the stinking bottle. Didn't? That's how it's been. We're way down the issue. All right. That's it for acids and bases. 3 minutes more. That's it. So we've already talked about molecules. We talked about bond. We made molecules and create or form those bonds. Right? Covalent. What's going on there? Those are what we call reactions. When we take two molecules and add them together to create something new, or we take a molecule and we split it apart to make two molecules. Those are what we call chemical reactions. We have reactants, that's what's being acted upon. We have a product, which is the result of that action. So we have again a few reactions that are three again. All about trinities. Three again. We have decomposition, we have synthesis, and we have exchange. Decomposition. Another word. Catabolic. Another word. Digestion. Another word. Breakdown. You take a molecule and what do you do to it? You break it down. Break it down. You decompose it. And it's called metabolism. Things are broken in smaller units. Synthesis is the opposite. So we're building, we're making, we're combining. We call it anabolic. Some of you are very familiar with anabolic. Nobody on the Asian football team would do anabolic steroids. Cross country runners do. No. Don't use steroids. Bad news. Now, synthesis, opposite of decomposition. What's the third? Exchange. In other words, we exchange pieces and parts. One of those three are the most important. The first two. The last one, we'll refer to it everyone. We really are going to dig in. Decomposition and synthesis. So, y'all went and had a wonderful lunch in the marketplace. I did. So, I was nom. And what was I doing? Was I doing decomposition or synthesis? Decomposition. All right? Now, I've absorbed some amino acids, absorbed a little bit of glucose. What am I going to do with those amino acids? Oh, boy. I mean, look at that. That's not too bad. I'm going to build. Okay? Or in my case, repair. You're going to be building things. Okay? So what's going on? Now, like 3 o'clock in the afternoon, I'm running out of energy. Am I going to go eat something? Good. I don't have to. What am I going to do? I think hopefully some of this stuff, convert it. It's stored. Let's break it down and use it. That's what this is. That's what we're describing here. Picture help? There's a starch molecule. That was the bread you ate. That's the Chick-fil-A waffle crust. Pizza crust. And what do you do with it? You break it down into glucose. Amino acids. Okay. I had cheese. That was about it. No, I had eggs. That's right. I have eggs on myself. So I had the eggs. That's got protein in it. I digested them. I absorbed the amino acids. Now I'm going to make protein. And there's the exchange. Notice again, that's it. All right? Now, most reactions, if not all, are reversible. I'm kind of describing it that way. I hope you understand. I eat starch in the bread. I break it down into glucose. I absorb the glucose. And then what do? I make a starch molecule and store it in my liver or my muscle. cross-country team, they have lots of nitrogen before they go run, right? Lots and lots and lots of starch. Build up, store in the muscle and liver for that 5K and whatever it runs. So these are reversible. Those can be built back into the starch molecule. Those would be built as protein. The protein can be broken down to that. Reversible. That's what we need. Now, how quickly does a reaction occur? Depends upon a few things. One is concentration of the reactants. Do I have a lot or a little? If I have a lot of reactants, hey, it's easier for me to bump into each other. Why so many people end up getting married after college? Come on, catch up. There's lots of people around. You're going to bump into somebody and have a reaction. Temperature. Which body temperature? How many degrees now?
38.6.
You don't say 37, you're wrong. 37 degrees?
So
No.
okay.
Remember what we use.
Yeah, yeah.
Everything you have.
I need it. Make sure.
If we change the temperature, then we change the rates of the reaction. Increase the temperature, what happens to the molecule? Move faster or slower when it heats up? Faster. You get better product delivery. Faster. Like when you got the fever, burn through some calories. And then there's something called a catalyst. We'll get to those later. All right. One last topic before we break for the weekend. Oh, that's not the end of the topic. I know that.
Like, that's what? I need to